to the Weekly Bioanalysis, a KCAS podcast.
Hello, and welcome to the 79th episode of the Weekly Bioanalysis. This is officially episode 1 of season number 3. We've been doing this so long that we're bona fide with 3 seasons. As I mentioned, this is the 79th episode of the official podcast of KCAS Bio. KCAS Bio is a bioanalytical CRO serving the pharmaceutical and biopharmaceutical industries for over 40 years. My name is Dominic Moreno, and I've been in the bioanalytical LBA and biomarker space for over 30 years. I'm here with my co-host, John Perkins.
Hi, I'm John Perkins, and I've been in the bioanalytical LCMS space for over 30 years.
John and I are members of the growing scientific advisory team here at KCAS Bio. Either or both of us are available to answer any questions you may have regarding this podcast or any of KCAS Bio services. We are amped up to have you listening to the 79th episode again. Unbelievable, 3rd season here. The podcast is now available virtually everywhere. Wherever you choose to find and play your podcast, you can now likely find the Weekly Bioanalysis. We welcome all of you, whether you're joining us for the first time or you're a regular listener. Today's podcast will be a review of the latest news and resources, and then a focus on a topic of our choosing before discussing any feedback we've had from you. We're constantly looking for topics, and we'd be happy to discuss something that you want us to cover. So again, we're amped up to have you here, and we are looking forward to a fun episode today. To kick us off for podcast number 79, again, officially episode number 1 of season 3, John is going to cover today's podcast a bit before we go and jump into the news and notes section. John?
I'll be absolutely honest, it's a little bit early in the morning for me to be amped up about anything. I'm not a morning person, but I'm doing my best to get going here. So this week, the main topic, we'll do the usual news and resources, but actually a lot of the news items feed into our main topic, which is the coming of age of one-and-done therapies, the present landscape of gene therapy / editing for non-malignant conditions. We're looking forward to it because it's actually a fascinating area and has a big impact on us as a business.
Great, John. Let's jump right in.
Sure. Yeah, it's all about genes today. The first one is scientists just discovered a new genetic clue for delaying Alzheimer's disease. It's already inspiring drug discovery. There's a family in Colombia who basically, it's 6,000 members, and nearly 1 in 5 carry a genetic mutation that causes amyloid protein to build up in their brains at a young age, causing cognitive impairments in their 40s, dementia in their 50s, and death in their 60s. So they basically have a very nasty and aggressive form of Alzheimer's, and there's one family member was identified early who had 2 copies of a protective mutation dubbed Christchurch, and whose disease was delayed by decades. And there's also a small number of family members who have a single copy of the mutation. So the theory was that you needed both copies to have protection. But as they looked at these family members who have the single copy, there was actually a delay in their Alzheimer's too. And the excitement about this is knowing, so when you're developing a drug, to have 100% effect is really difficult to reproduce, but 50% effect is a reasonable target, and knowing that only have one of these mutations is sufficient to cause an effect, that's driving drug discovery in this team at Oregon Health and Science University. Let me say that again. I know I'm really sorry about this. It's just first thing in the morning. Again, I'll step back. So I'm sorry, Jeremy, but we'll give you a bit on this. So basically, looking at brain scans of people with these mutations, they found that they had a lot of amyloid plaque, but very little buildup of tau, so there's a lot of discussion about what's responsible for Alzheimer's. There's obviously been a lot of, amyloid plaque was pointed to as the main source, but this group are actually thinking that the tau pathology is actually is actually key to possibly addressing Alzheimer's, and they then looked into the path of what prevented build up of tau. And I'll pass it on to Dominic at this point, and I'll be ready to
Yeah,
talk.
it's a this the brain is literally a mesh of, or a network of like meshing that is still being elicited in terms of what exactly happens. Too much of a protein, too little of a protein, John. That's the biggest problem with. So some neurodegenerative disorders have a lack of amyloid. You mentioned it, tau, or too much of it, but if you don't have the right amount, it is as debilitating. And some forms cause a reduction, some cause a buildup. Still trying to sort out some of that, but their primary the gene of interest is this ApoE,
Yes.
specifically ApoE2 and 4. Those are biomarkers that are that we've measured, John. In fact, this story has a lot of like germane, all those biomarkers we have methods for, tau, right, A
Mhm.
beta,
Yep. Yep.
ApoE4, especially 4. But it looks like the whole, and it's a fascinating story because I got this massive subset of patients, right? 6,000 people.
Yep.
Some have the mutation that protects them. Some are homozygous for it or heterozygous for it, or they have one of each, and that creates some sort of delay, right? And then basically, if I'm gathering this right, this is where it gets a little kind of hard to truly understand. They have normal amyloid levels in the, in the,
They have the expected buildup of amyloid, but
yeah,
it's the
but they
tau.
lack, yeah,
Yeah,
lack
and
in
if I
tau, right?
and if I step in,
You go
so
ahead.
this
Go ahead.
this group are the group is actually at the Mass Eye and Ear in Boston. So this is all in the New England Journal of Medicine. This was published. It's actually the mutation hinders the natural ability of ApoE from binding to proteins that are crucial for ferrying tau into cells. By restricting their interactions with ApoE, more of them are free to help microglia chew up and destroy tau. So the thinking behind this is actually ApoE is critical to all this. So the researchers, someone called Joseph Arboleda Velasquez, and his team designed an antibody that blocks ApoE, mimicking the effects of the Christchurch mutation. And they tested that in mice, and their thinking is that this is a good alternative target to go after for Alzheimer's. There's been a lot of focus on the plaques, etc., but ApoE's been largely ignored, and they're thinking this is a way to go at it. So again, for
Yeah.
them, it's early work. They're thinking possibly a CRISPR approach in the long term, where you're changing genes, may be a way to go. But it's the first step is to see if they can get an antibody that will have the desired effect.
Yeah. I mean, look, it's promising in a field that's had a lot of setbacks, right, John? There's been
Exactly.
lots of trials in this
Yes.
space,
Yes.
easily decade, if not decades of failure. We're
And,
probably borderlining on almost decades, at least two now of failed, say, neurodegenerative drugs. And now along comes this really interesting subset of patients, right? Let's start there. They can almost work back, not almost, they worked backwards and found this ApoE. Now, there's all these other problems with these disorders, and I know this has taken a long time, but I think it's to point out, right? Even though they're discovering antibodies against ApoE4, what's the problem is the blood-brain barrier, right, John?
yep.
Still
Yep.
something that people, there's stuff that can actively get across passively. We could talk a long time about just what I said around the blood-brain barrier, but that's the challenge with a mAb because ApoE4 is ubiquitously expressed. You don't want to mop it up all in the body. You just want to block it in the brain. So they're thinking about giving it, somebody maybe cranial. But that's why you mentioned CRISPR. This is a prime target for something like CRISPR where you could get more targeted or personalized medicine. So really it's actually, no, it took a little while to get through, John, but it's the first story where we've got that subset of patients that has the genetic background there to help to move things. So I don't know. It should be it looks very promising, at least as a way to continue to battle neurodegenerative disorders, which I think we all should be somewhat fearing. But then again, I look at it as at a certain point, John, I'm okay. No matter, if I get past a certain age, I'm going to be happy, and I'm getting there. So, but I don't know if you want to add anything. I hope that helped. But it's a
Well, no,
really interesting story, and one that
it's
has some promise for sure. It definitely does. Yeah.
yeah, it's really interesting. Just I'm a little bit disappointed at my slow start
Yeah, don't
here.
worry about it. Yeah, yeah. If those that are listening, we're a little bit unusual here. We're starting really early in the morning because of sun hours and various things. But we've got a real early start, and both John and I are, still trying to get awake. So it's all right. Our audience doesn't mind, John. We can.
I would
They,
never,
they, they understand. They feel our pain. So
I'd,
thanks for
ne-
everybody out
I'd
there.
never convincingly describe myself as a morning person. That's for
Yeah,
sure.
I actually am. So I feel like I'm a little ahead of you, but that's okay, John. I love the mornings, even though they're getting harder to love. But yeah, no. So great story there, John. Way to kick us off with the
So
first of
let's
many
move on.
interesting
So
articles here. Yeah.
this is obviously another topic that's been in the news for decades. A seventh known patient has been cured of HIV. Is the headline. It's basically an adult man dubbed the next Berlin patient has been declared the seventh person to be cured of HIV, and his case provides valuable information that could lead to a more broadly accessible approach for the 39 million people living with the virus around the globe. I mean, obviously, a lot of the drugs that we know about really are just about keeping it in check, but not. It's not. None of them are a cure. So, that there are daily regimens that people can take and largely keep it under control. But this is the patient was diagnosed in 2009 with HIV, then later developed acute myeloid leukemia. So then that patient needed a hematopoietic stem cell transplant in his bone marrow to treat his cancer. And so the team started looking for donors with a rare genetic mutation known as homozygous delta 32 CCR5 mutation, because that this mutation provides natural resistance to HIV. So a little bit of background: CCR5 is a receptor protein on white blood cells that HIV uses to infect cells with the delta 32 mutation. The virus can't bind to the protein and enter the cell. As a retrovirus, HIV then inserts part of its DNA into the genomes of infected cells, turning a reservoir of viral material in the body that is tough to eradicate. So they couldn't find a donor with two copies of protective mutations. So there's a bit of a theme here today, but did find someone who was heterozygous, meaning they had one copy of the gene with a mutation and one copy without it. They went ahead with the transplant and found that not only it treated the patient's cancer, but also seemed to cure HIV. And that a number of the other patients who've had this again cure have gone with the same path, where they've had leukemia, then they've needed a stem cell transplant in the bone marrow with the same approach, and it's actually been longer term rendered them HIV free. And so it's this again, the key here is that need for only one mutation seems to be actually sufficient to have the curative effect. I'll
Yeah.
pass it back to you.
Yeah, again, it's a really interesting story where people are HIV positive, right? They end up people live in long lives, as you've mentioned, but it's still kind of latency, right? And then they get leukemia, lymphoma, and they get treated for that with a bone marrow transplant. And lo and behold, they start becoming like long term. This isn't a short term thing. This is
No,
a long
absolutely, yeah,
term
yeah,
negative, all due to these newer, so mutated CCR5 cells that don't allow for the virus to uptake. And as you kind of indicated, it didn't even have to be a homozygous
yeah,
donors; it could be heterozygous. And then there's actually one patient, right, that didn't get any of these mutated gamma delta that is still negative, although it hasn't been as long term. So
yeah,
I think
yeah,
some of it is just getting newer cells is good, John. Right?
yeah,
You've kind of heard me jokingly say I'm going to take my own bone marrow out and treat it with IL-4 and GM-CSF and create my own dendritic cells and put them back into myself. I think that would be beneficial. I want to stockpile them and inject them in my face if I can. No, I'm just kidding, John.
sure,
So no, but like I hope that made sense, right? Like this is the therapy. It's clear there's some benefit to these
yep,
cells, but
and
even the therapy itself does, and more importantly, I think we're getting so close to a cure in some capacity. It's fascinating stuff. There's got to be a
Yeah.
way to understand that, and then instead of having to go through a whole transplant, there's probably a way to, I don't know if we could CRISPR, mutate delta, the CCR, the delta 32 gene, right? That's ultimately what the therapy could be. But I'll give it back to you, John.
So
This is unbelievable, right? This is like 30, 40 years of science coming to fruition now.
sure, yeah. Just a couple of key takeaways. I mean, the fact that it doesn't have to be homozygous means that you've got a bigger pool to pull from donors. So that's more optimistic in itself. So heterozygous donors are at 16% of the population versus 1% who have the homozygous mutation. However, one piece of this is these bone marrow transplants aren't something that you'd routinely want to do. So it really isn't feasible to just say anyone who has HIV can have a bone marrow transplant, because obviously that it's not a pleasant procedure to go through. So it's all about this finding how can we take advantage of the fact that the one mutation is sufficient to possibly cure it, and then that gives a route to hopefully address the disease in the long term.
Yeah, no, well said, John. It's fascinating. It's certainly not sustainable to give all 39 million people
No,
with
absolutely not.
Yeah. So that is where, what else could we potentially do? And I still think there's some things to be unlocked in there. Undoubtedly, I do think this mutation plays a part, but in general, just getting somebody, it looks like clean central memory cells may be beneficial. But let's maybe we can move on to the next article here, John. But great stuff there, both.
I quite like this little bit of research. Well, it's not a little bit of research. That I'm not diminishing it at all. This is the neural circuits behind the placebo effect, and it's interesting where you'll be reading about drug trials where a drug's in phase 2, phase 3, and it fails because the placebo patients are shown as much of an effect as the actual experimental therapy. And this is a problem particularly with pain drugs, where you have this placebo effect, and it's trying to figure out why does that happen. And this is a group at the University of Medicine Essen in Germany, who are looking at. Oh, sorry, this is not. I've done it again. This is not at Essen in Germany. This was actually. It's. Hold on a minute. I'll have to tell you what the group is. Let's step back a bit. So basically, we have the issue with placebo effect. Particularly in pain drugs, where you've got placebo provides relief to make clinical trials fail, but it's trying to understand why do brains produce pain relief through belief alone, and what's the region of the brain that leads to the placebo effect, again to hopefully drive the understanding and figure out what can we do in terms of drug development, and is that sufficient, or would that help with development of more non-opioid based drugs? We talked about the Vertex drug recently that's obviously going through late phase studies, but the more we can develop pain medications that don't have the addictive side effects, then that's a good thing. So this group actually looked at mice, and they started off with basically the mice had two chambers. One of them was heated the floor of the chamber is heated really hot, and so the second chamber had a cool floor. So the mice realized that if they went from the hot floor to the cool floor, they could relieve the pain in the feet, and so they got the mice conditioned to this kind of condition. And I'm not sure how long they did. Just say they basically, let's say a week or so. I'm not sure what the timing is, but they took the time to condition them to that. Then the next step was actually they put both chambers had hot floors. So when the mouse was in the chamber it was used to being hot, got its hot feet, it jumped to the other chamber, and then that's where the placebo kicked in. Placebo effect kicked in because it was conditioned to think that floor would be cool, even though it wasn't at all. And so basically, what they did was they had miniature microscopes on the mice's head, which revealed where the activity was in the brain when they were experiencing the placebo effect. And they were seeing it in the rostral anterior cingulated cortex, and which is a brain region known to be involved in pain. The group expected the amygdala to light up, but that's not what they found. They found instead that neurons that connect the rostro anterior cingulated cortex, that connected that to a part of the pons called the pontine nucleus, showed the most activity. So located down in the brainstem, the pontine nucleus leads only to the cerebellum, a neighborhood in the back of the brain best known for coordinating movement. So this was basically flagging bits of the brain that have never been thought of as being involved in pain at all. So the, this was basically, it was this really interesting experiment showed that the brain, again, it's given us more understanding of how the brain reacts to things than had been thought of before. I'll pass it back on to you.
I mean, it is a fascinating story, right, John? Like, just to come up with this type of experimentation to kind of create
Yeah,
the placebo effect in and of itself is really, really cool. And then to mount a camera, right, a mini microscope
yeah,
onto the
sure.
heads of the mice to see this, this rostral anterior cingulated cortex, RACC, right? But like, John, I'm fascinated by this story because we've always talked about placebo effects, and at least I'm a firm believer in, belief is a powerful thing, John, right? If you
Sure.
believe you're sick, you can actually manifest it if people have willed themselves into health that way. Now, I'm still saying you should go see the doctor and take medicines. Don't just sit there and, I love the body by chemistry as I'm entering my, getting into my 50s, right, John? But the thought of this experimentation where you can now physically see the mouse their beliefs are so strong that they're safer on this side of the chamber, that they've really relieved their pain. That is amazing. I guess my mom was right when she said, just think warm, when I was standing out in the New England cold, right? I guess that's what it comes down to. She was right.
But yeah, I mean, you're right. It
But
can be
it's the
really,
same thing, right? When you think yourself warm,
yeah.
think cold. It's the same concept here. It's really fascinating.
It is amazing what you can force yourself to do.
Yeah.
I was just, while you were mentioning, I was thinking of an experience I had ages ago, where I actually had tickets to go and see Doves down in Philadelphia, but 2 days before I developed an abscess in my tooth, and it was majorly painful. Um, but I actually drove down to Philadelphia, saw, and it's a 4 hour drive either way. Saw the band, drove home the next day, and then just collapsed. And I was laid out for the next couple of days. But is that? I mean, you've got that mental power to do stuff.
Yeah.
I mean, I was a mess when I got back, but somehow I got myself, to it and
Yeah,
through it.
no, it is like that. That's you just described how and I guess my stories are more no pain, no gain. That used to be the mindset. I don't know. Maybe I say used to. I don't work out like I quite used to, John. But like every day, like it was no pain, no gain in the gym. So if you weren't hurting the next day, it was a bad thing in my world. So I don't know, John. It's a great story though, and
So anyway,
yeah, we can
just
keep
to wrap
along the theme here. Yeah, we
Yeah,
got a good theme.
just to wrap it up, basically, yeah, like I say, this is all about thinking if we target the pons and cerebellum, then we might be able to come up with more non-addictive pain
Yeah.
management.
Oh, definitely. Yeah.
Um,
Yeah.
and so
Yeah.
the next, sorry, the next step for this group is actually to see if this works in humans. This was mouse, so it's a question of what can we do with humans, and
Yeah. Stay tuned with that
whether
pathway, right?
this works or not.
Yeah.
And I'm going to let's see. Uh, it's so basically the group that did it. It's a guy called Gregory Scherer at University of North Carolina in Chapel Hill. That's I always like to acknowledge who does this work, and I couldn't catch it right at the beginning, but that's he was the main researcher on this.
Yeah. No, it's a great story. There's definitely stay tuned there. There's some interesting pain things that'll be associated with RACC as well as the pons, right? The prawns, the pontine nucleus pawns, right?
Yep. So we'll move on, and yes, let's say it's all genes today, because this is actually the biggest news in probably the last however many weeks is scientists unveil new way to make large changes to genes and no CRISPR is required. So this is science. I'm going to read a lot of this because it's
Yeah, it's
quite
a good
frankly
one.
it's over my head. Scientists led by Patrick Hsu at the Arc Institute in Palo Alto have unveiled a new form of gene editing that can add, remove, or flip large stretches of DNA, all without the help of CRISPR. So basically, just as a bit of background, so CRISPR gene editing ushered in a new era for biology just over a decade ago. An easily reprogrammable so a guide NRA. Sorry, I don't know what has happened to my language here. Guide NRAs are key to the versatility of CRISPR, but CRISPR tools are best at breaking problematic genes or making small edits. Inserting whole genes or making sweeping changes to existing ones is still hard and is increasingly the focus of several new biotech startups. So this, like I said, the team at Palo Alto, they rather than using guide NRA, they have a different approach called what they've called bridge RNA. It's similar to guide RNA, but recognizes two stretches of DNA at once, the target site for editing and the new gene to be inserted. In forming that bridge, it recruits an enzyme called a recombinase to perform the actual edit. This allows us to specify any two DNA sequences of interest that we want to combine. Hsu said we can insert DNA, but we can also delete it or invert it with this new system in a universal mechanism. And I'll pass it back to you because this is
Yeah,
much.
it's good. It's hard to understand some of this if you're not in the space, John, and it's an evolving
Exactly.
space, right? This
Yep.
is still, this is still. I guess it's starting to gain a lot more traction, but you kind of nailed it. There's these. They're dubbed a bridge RNA versus guided RNAs. And so in the past, the CRISPR, as you mentioned, were guided RNAs, and they had to take advantage of the machinery of the cell, but they had to make cuts and breaks and so forth. This can do it where there's any double-stranded DNA, I think, and it forms a bridge. And John, I got to tell you, it's still a little. So this kind of brings on a third version of this stuff, is what they're saying. And when I say it that way, it's all John, I think it's hard for us to understand in part because I think they're still a little unsure of you can't get in
Mhm.
there and visually watch the stitching, right? It's not like a sewing machine, kind of
Yep.
think of it that way, where you can see where, they're cutting out this and insert this, right? It's all still done in like a molecular fashion, right? So it's a little bit hard for them to kind of lay out exactly. They can show, hey, here's what we think it is, but again, it's
Yep.
still a little bit hypothetical. How's that, right? I guess
Yeah.
that's where I'm
Absolutely.
going
Yeah. That's
with it to kind of cut us a little bit of a break because neither of us are gene jockeys, right, John? We're not molecular biologists. At least I've dabbled in it. But anyway, I digress because it's like I think what's really interesting here is you mentioned the recombinases and it's a new and very unexpected way to do this. Instead of the guided, they can bridge it. So I think instead of like kind of trying to target the mutated DNA and stick in there, these just form when they see these anything that's in a is a they can target any sort of two DNA sequences that they want, right? So it's a little bit it's really fascinating, John. And I think what I'm trying to say is it's the third wave of RNA guided systems following gene silencing with RNAis and gene editing with CRISPRs, right? This isn't really editing, these are bridging,
yeah.
right? So you got
I think.
silencing,
I think the bridging.
editing, and bridging. That's
I,
kind of
I,
the three
I,
buckets.
but I think it's bridging and editing. I think. Again, I'm trying to get my head around it. I think
And it's
there's a
only in
couple.
bacteria, right? So they still haven't, this
Yeah.
is still all
It's
really new stuff. So
Oh, it's
yeah,
really, really early on. I think this is there's a couple of paragraphs here. It says science have used recombinases as a research tool for decades, but the enzymes are rigidly set in their ways, only targeting specific stretches of DNA. That is simple means to control where they make their edits. They never took off as a starting point for new medicines. So what this does is the bridge RNA can control recombinases, um, so then to actually direct them where they can make these large genetic changes.
Yeah,
Is how I
yeah, John, but
understand
it's
it.
not like they're sitting there keying it into a keyboard and telling it where to go,
Oh no, no,
right?
no, absolutely not.
You're
Yeah,
not like playing in, like a joystick, right? This is
yeah,
all Brownian movement. Let's call it what it is, right? There's still a system of, stuff kind of making their way, and they're just enzymes that are kind of thumping along. Oh, doing nothing. Oh, I hit the right area. Boom! I'm gonna make this bridge. It's still great stuff. A boom is a technical term, John, if you're wondering.
yes.
Yeah.
So, I don't know if there's much more to say in this. I think because, like you
No,
said,
it's so early on that this is still in bacteria, but stay tuned. There's a third wave of gene editing now. I guess we
that's
shouldn't call it
that's
editing, but yeah, there's a third way of, third class of sort of gene therapies here after the silencers, the editors, and now the bridgers. It's great
yeah.
stuff.
Like I say, it will be coming. Yeah,
Yeah.
that's the thing. It's early days, but yeah, well, I'm sure we'll hear more about it as it moves into mice and larger.
Well, John, I think our news and notes ties in nicely to our main topic today, doesn't it?
Exactly. Yeah, yeah. It's all about genes. Like I say, it's all about genes today.
Yeah.
So the main topic this was an article. It was actually, I think it's William Blair who are like investment guidance, etc. And this article was just it was called "The Coming of Age of One and Done Therapies: The Present Landscape of Gene Therapy Editing for Non-Malignant Conditions." And it just seemed it was an article that jumped out to both of us, and we've had it sitting on the back burner for a while. But because obviously we have groups at KCAS who work in this area in terms of providing bioanalytical. It made sense to have this as a topic when we thought the time was appropriate, and given all the news items were all talking about gene work, etc., it seemed like a really good way to tie everything together. So I'll some of it will be just reading because again,
Yeah.
this is not
Okay.
my area of expertise by any means. But I thought there was a couple of things in that article where it basically says gene therapy is broadly defined as the introduction, removal, or change in genetic material to treat or prevent disease. The genetic material can be packaged into the delivery vehicle and directly injected into a patient to enact its therapeutic effect, or introduced into cells that have been removed from a person with the intention of reintroducing the modified cells back into the patient. While the majority of gene therapies are intended to be one and done therapies, some you need additional treatments. But this piece talks about all the different types of gene therapies that are out there, either replacements or gene alteration. And then another big piece for us is the delivery. Is it AAV delivery? Is it lipid nanoparticle delivery? So there's a lot of focus in the field on that,
Mm.
and it's just it's just a nice overall summary, and we'll try and talk our way through it, and then touch on where KCAS can help companies who are in this arena.
Yeah. No, John. You're there's some. I mean, where to begin? This is a massive. We're not going to be able to cover all of this in the next 30 if we want to kind of cap our discussion today, because there's just. Let's just dig in, right? I think there's some unbelievable stats. Or do you want to start with the type of therapies? It's your
I think
call.
I think maybe it is worth saying among the ones that are out there, really the type of therapies do sort of make sense. So you've got gene replacement, where you're adding a new gene to a cell, new functional gene added to a cell. Double-stranded knockout/knockdown, which is like a double-stranded break, DNA repair, site-specific indels, downregulation or no gene expression. You get insertion of gene insertion, gene excision, which is removal of a desired gene and DNA repair, epigenetic, which is for upregulation and downregulation, base editing, reverse transcriptase, and LV integration. So again, for some of this, for me, it's a foreign language. But I think if you want to talk about any of these, feel free.
No, yeah. So you just mentioned nicely the 2, 4, 6, 8 really categories of gene replacement and gene editing modalities. And yeah, again, you could go a whole hour on just trying to go through the 3 of them, but it's you kind of you either replace them, right? You knock them out, you insert them, you excise them, you upregulate or downregulate. You can do whole swaths of single site mutation or changes or big stretches of changes, and then some of it has to do with like if the you target DNA and RNA. That's kind of and then the integration of the viral delivery of the RNA, which is really one of the more exploding fields, right? That last one you talked about, the LV
Sure. Yep.
integration. But I hope the audience gets a sense that there's just a whole different way of tackling this, and a lot of it's based off of the disease. That's kind of how you're going to go after it. But no, just switching, just kind of following along the theme, John. I think it's important just to set the foundation here. There's 677 gene therapy or editing programs from 210 companies, right? Let's let
yes,
that settle
exactly.
in first.
I think that's key. That's exactly where I was going to go next.
667 and 210 companies, and you can pick it up from there. Like they say, 36% of the companies are publicly traded, but that's a lot of research going into this space, right? It was almost surprising to read 210 companies, kind of in this gene editing or how kind of gene therapy space, and probably I wonder how they even got to that number. Meaning, are we talking about? I always talk about cell-based gene therapy companies, right? Those are your CAR Ts and all that.
Mm-hmm.
Or are we talking about more traditional, it's not even traditional, it's just gene therapies where you've got some sort of RNA, could be an ARC, right? Now companies that are ARC companies are technically gene therapy companies. You see how quickly they went from biologics to gene therapies, right? Because you're adding that inhibitory RNA as part of your protein. So the field is booming. So I wonder if that's even a little underreported, John, is what I'm saying. Because then you got traditional LNPs and oligos, right? Let's just set the stage of what we call gene therapies is growing, right? In the past, you might have 10 years ago, someone said gene therapy, you probably thought, oh wow, that's a really complex, got to be this. Now it's like there's like 6-8 different categories that are falling into gene therapies, like that were traditionally more just, you didn't think of an ASO as gene therapy, but it really is, right?
Sure.
And so are a lot of LNPs, and obviously, cell-based gene therapies are pretty obvious, right? Or anything, if someone's treating just with some sort of viral vector, oh yeah, that's a gene therapy. But now it is moving into peptides, right? It is moving into, but not peptides, but potentially into oligo space, as well as obviously anything that has a silencing RNA is now a gene therapy. Or sometimes you're knocking it back in. So little side note there, but just it's a huge field, right, John? So you got this massive field, you got 6 different ways to tackle it. And what's the holy grail, right? It's that one and done concept here.
Exactly. Yeah.
It's amazing how effective some of these things are. But yeah, I'll put myself back, or I'm going to pass it back to you, John. But
that's fine. I mean, it gives me a chance as we bat it back and forth. Gives me a chance to dig into it too. So, like you say, according to this report, there's 677 gene therapies in development that they identified. The bulk of them are gene replacement approaches, and 70% are gene replacement, 30% are gene editing approaches. AAVs continues to be the most widely used gene therapy modality, while lentiviral-based integration and other methods for gene replacement are less common. It's interesting that the gene replacement is more focused in neurology and ophthalmological indications. Gene editing is more focused on hematology, cardiology, and neurology / neuromuscular indications. Again, when you say there's a lot of these in development, the bulk of them are still in early development, so it's still in preclinical. It's 78%, well, 17% are in early clinical development. There are 22 programs in late stage development, and I'm not going to go through the names of them, but of course we have seen approved medication. So this is a field that is rapidly maturing, because we are getting drugs to market. It's just it's a lot of it is about how do we most efficiently deliver things like that to get to where we need to be, to treat the condition that we're aiming at.
Yeah, but it's interesting, the whole breakdown here of 70% of them are in the gene replacement and 30 in the editing. So that's an interesting first swath at it. And then I like how they give you the indications, right? You talk about neurology, neuromuscular, ophthalmological, right? 22% almost split. I was a little surprised to hear about how
Yeah,
much of this is
yeah.
going into the eye, right? That's probably the first thing that stuck out. And then when
But
they say,
I
yeah, go
do
ahead.
know that we've worked with a
Yeah.
couple of companies who are doing where it's they're basically it's the injection into the
I think
eye.
all actually
Yeah,
now that you say that, John, the maybe half dozen that we're working with are all doing gene
yeah,
therapy in the eye.
yeah,
So
yeah.
it makes sense now, but of course, retrospectively, I would have thought the neuro would have taken up probably 2/3 of it, right?
Sure.
When the
Yep.
reality is it's only
Yep.
22%. I think the hematology one is probably where I don't know what that's probably leukemia, lymphoma type therapies, right?
Hmm.
So that's 16%. It's the cardiology at 14, um, and so. But anyhow, it's an interesting breakdown, and 14% are undiagnosed. They don't really tell you, which I think is even
Sure.
more
Yeah.
like,
Yeah.
oh, what does that mean, right? What does that
There's,
mean?
there's some cagey people out there still that keep it
Yeah,
under
they're
wraps.
they're they're in they're in stealth mode. They're holding on to
Yes.
that
Yeah.
as tight as possible. But I mean, just in general, do we want to give a shout out to some of the diseases that are being treated in the companies, or do we want to move on?
Have you do whatever you want? I don't.
Yeah, because I
This
think
is
it's a lot of it are in the ALS and the Duchenne's, right? But DMD, hemophilia, Parkinson, Alzheimer's are all really a lot of what they're targeting. So there are some. Sarepta is one that has
Yep.
a dystrophin, the Duchenne, excuse me, Duchenne's, and then so that's we've heard Sarepta is a big player in the space, and their DMD, right, the Duchenne's muscular dystrophy therapeutic is life changing, right? It's unbelievable. And then BioMarin is another um, renowned company with their hemophilia A. Well, we're very familiar with that. And that's these are all one and dones, John. By the way, right?
Hmm.
Those are the
Yeah.
two
Sure.
big ones
Yep.
that are
Yep.
one and dones and approved. And then you got some. Well, maybe I'll take a pause there. There are a few others for like Bluebird. They mentioned them, and then CRISPR themselves, right? The company CRISPR is out there. So those are really some big players. Those drugs are treating gene editing. Those are the leading gene. So the ones I mentioned before were the gene replacement leaders, and these are the gene editing ones. But and that CRISPR Vertex Casagevi. I don't know if I'm
It's
saying that
heavy.
one
Yeah.
right.
Yeah. Yeah.
Yeah,
Yeah.
that one's again a really life changing drug, John. That one's
Sure.
got a lot of
Exactly.
momentum to it. So
And that.
I'll take a pause there. But there's just it's I guess I didn't really fully grasp the editing versus the replacement fields and just how important that is. But boy, it's coming at me like a fastball this morning. Pretty cool stuff.
Yes, I think, and this is. I mean, one of the factors of these drugs is because they're one and done, their price tags are quite eye-watering when you
Mm.
see them. So I know, particularly in Europe, where you've got the medical regulators are a little bit more price conscious than here, there is a fair amount of pushback on adoption of these therapies. So I think there's concern from the investment community about, well, what's likely to if you've got a gene therapy, is it? Yeah, it might get to approval. How's it going to succeed? And I think it's the thought is that you have to the ones that are most likely to succeed for the companies are those that are treating indications that are fatal in childhood or have a high morbidity and no disease-modifying therapies available. I think at the moment there's still a number of you hear multiple companies going for the same indication, like Duchenne. There's a lot of work being done in that area in terms of gene therapies, but also it's still early days. That you hear a lot of discussion and things fall by the wayside. Because I think there was the Pfizer Duchenne one that I think had major problems recently with side effects and treatment. So I think it's still early days in figuring out where this field's going to go. But this was just, like, say, one of the assessments is if you've got an indication that has no approach, or no other therapy available, then there's a strong indication that if your therapy works, then it should do well out there.
Yeah, no, and I think you mentioned that price tag and efficacy and all that, and there's some nice nuggets to kind of chew on here. One is, in general, the breakdown of the delivery is really going to impact the price tag and efficacy, right? So there's 496 programs that identify as viral-based delivery. So these are your AAVs, your LVs, and a lot of people use, and they even give you a breakdown. Some are herpes, HSVs. So your AAVs are 63%, the adeno-associated ones, right? Your LVs, the lentiviral ones, are 6%, and those HSVs, which are the 2%. So that's the delivery. Think about and some other delivery systems are viral-like particles and aneoviruses, which that one I'm not really familiar with. Maybe they're saying anal virus. I don't know what that says. I'm not familiar with this other viral form. But let me just, of those 496 programs, AAVs are 63%, LVs 6%, HSVs are 2%, and then others are 6%. And those are those virus-like particles. Why is Dom talking about this? Well, those are traditionally not one and done, but usually there's might be a couple shots of these, right? Think about how you get a HPV vaccine or an HPV vaccine, but still relatively affordable to make, have good, easy to deliver. But I don't know, like the efficacy of them is where you might need multiple injections, and even treating some of your more, let's call them challenging disorders like Duchenne's and some of the things we're talking about, they haven't really had quite the efficacy, at least to the best of my knowledge. They haven't, or maybe they have, but certainly that's a large swath of the 667 therapeutics, 496 in this bucket, right?
Yeah.
But then they kind of break it down, what they call these are 11 which use the lentivirus system that are ex vivo gene therapies, and this is where we get into those cell mediated gene therapies, right? These are the big companies here. There's Bluebird Bio's on here, Rocket, right? If you've heard of them, Kyowa Kirna's on there, and Mustang Bio fall into these. And these companies are all the LV space is growing, but those are a little bit, those players have been going at it for a good 10 years, most of them on there, or say 5. But that's just an interesting breakdown between the large number that are using just, well, I don't even like the word traditional. They call them viral or non-viral delivery techniques for gene editing. And then there's the ex vivo gene therapies. And you talked about price tags. Well, the ones that are viral or non-viral delivery, they're still charging a healthy amount, but they're easy to make. It's those ex vivo ones that the price tag's enormous, and typically it's just a single injection is what they want to do there. So I don't know. You want to add anything there, John? There's more. They give further breakdown. Only 16% of the programs using non-viral, so that's 132 in vivo gene editing candidates, and half of them are using LNPs as the delivery. So we're getting into that space, right? So I think that's the next bucket, right? If our audience is trying to follow me, there's buckets of just your true viral based deliveries, the AAVs, the LVs. HPV things, your ex vivo gene therapies using LVs, and then you're getting into the LNPs of the world and other sorts of ways to bring those things in. So John, I'll pass it back over to you.
I was going to.
It's really cool.
I was going to jump in the lipid nanoparticles because this is actually a really interesting section. So, a major focus of the field has been the development of non-viral delivery technologies that are more scalable, they enable redosing, and do not require the use of concomitant steroids. So they think that William Blair's assessment is that the landscape will shift more towards these non-viral delivery options, and there's a number of companies who are working on lipid nanoparticle approaches and actually trying to develop proprietary LNPs that have improved tolerability or can target tissues other than the liver. That's one of the things with the LNP approach. They currently they go to the liver as the target organ, and then the therapeutic is, I assume, released from there. But companies like Poseida, CRISPR, and Beam are all looking in this area as to looking at the technology, the lipid nanoparticle, as to how can you improve that to better improve the efficacy of your medication.
Yeah, it's a. I mean, we could just keep going on and on here. There's so many other breakdowns of just where the market is in terms of gene therapies. Yeah, John, you mentioned the traditional LNPs. Um, then we kind of, those are all like um, the ones we kind of touched on are more vaccine type deliveries as well as ex vivo kind of therapeutics, and then they shift gears into the in vivo ones, right?
Mm-hmm.
And that's an interesting space too if we want to head down that path where there's 580 programs in this space, but only 47% don't even tell you the route of administration, John. That's how proprietary some of this stuff is becoming, right?
Oh, sure.
That
Yeah,
was
yeah.
fascinating to read. And then they talk about intraocular makes up the largest bucket of administration. Back to the eye,
Yep.
and then intravenous infusions, and then some intracranial, right? Intrathecal, intrabrachial, ventricle. I mean, this is where it's getting, or intracisterno magna. I'm not even sure I know what that is, John. Right? So, but that it's some brain regional type stuff where you're really getting sophisticated with the route of delivery. But that is what it's going to take to cure some of these
Oh,
debilitating
sure.
diseases.
Absolutely. Yeah.
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 And so that kind of rounds out a little bit there. I don't know where you want to go because I think we've done a great job of
I,
going over our agenda here.
I'm not
But
sure. I don't have a lot more to say on the actual
yeah,
gene therapies. I wonder if it's worth saying. Well, I don't know if you have. I mean, you're closer to it than me. Do you know? Have you any thoughts beyond this where the space is going? And then I suppose the biggest deal is where the kind of services that we can provide that actually we can support this kind
yeah,
of work.
John, this is why we set up some news and resources to go over where it is going. It's going to be these
Yeah,
bridging
sure.
ones, right? That is,
Yeah,
we
yeah,
kind of, you talked about it. This is when if you think about when we started the podcast now, I don't know, X number of years ago, and we are in our third season, episode 1, right? So when you think about when we started, we were still, CRISPR was just palindrome repeats. We were trying to figure out what the hell it meant, right?
yeah,
Now
yeah.
today we're seeing it in the therapies, right? There's some movement in it. It's accelerated, right? Seriously, science accelerated it. We're bombing relative to where we were 20, 30 years ago with the advancement of these new technologies. Well, that's it, the bridging, right? No more. These are much more I don't know, but maybe we'll talk a little bit more about the differences. That might be something good to do. Maybe we bring on Carrie and we'll talk about that in a couple weeks or a couple episodes from now,
Sure.
because
Yeah,
I do think it's a great question. Where is this field going? Well, it seems like they're going away from that gene editing with CRISPR, right, to where they called it guided gene
sure.
editing, to this bridging gene editing, which is great. I mean, it's definitely. And then it's just to kind of switch gears when we look at the cell based gene therapies. They're having, there's these side effects that people are worried about. Although it's low, it's rare, meaning maybe 1 in 30,000, but it still has this perception of being
Sure.
some side effects from these therapeutics. So what are they doing? They're making bigger, better, badder ones, trying to, whatever sort of LV or AAV they're using, they're trying to figure out ways to make that The next wave of CAR Ts is to make them less immunogenic, John, is where I'm going. So
Sure.
they're already starting to try to combat that, to work on that kind of word that's been coined, organogenesis, right? So some of the CAR T cells are curing maybe the immuno-oncology one specifically, they're curing the cancers that the patient has, but they are, maybe the side effect is it might be causing some sort of sprout of a different sort of cancer type. And so there's an advancement there. They're looking at ways to say, hey, what is the side effect that's causing that, and can we tweak it to remove it? And
Sure.
that's
Absolutely.
awesome too.
Yep.
Say, the whole AAV field, I could go on and on, John. The AAV field itself has made major advancements with overcoming pre-existing antibodies against AAVs. That's why it's not a worry. That's why it's not a mandatory test for inclusion exclusion anymore, because people are creating AAVs that all the pre-existing stuff out there is not neutralizing it anymore, John. Or they're taking different pieces of AAVs, and now they know whether it's AAV9 or 2 or 7, what immunologically, oh, this generates these antibody types, oh, this causes this unwanted immunogenicity. So they're grabbing pieces of the AAVs and cannibalizing them and creating like Franken AAVs, right? They're like just parts of AAVs, so that they evade and become more potent in their efficacy and less immunogenic, less toxicity, and safer. So that's a
Sure.
huge
Yep.
movement there, right? And you hear clients coming to us now with, oh, it's part this one, part that one. That's a major kind of advancement. So I mean, hey, I could keep going, but I'll give you a little bit of flavor of things behind it. But that was a great question. I love it. The field is, there's no, and I bet you if we did a little bit more searching, there's probably a handful of new waves of even the next CRISPRs, right? And we kind of touched on with these
Sure.
bridging RNAs. That is the wave there. But I assure you, there's a whole probably slew of other sort of technologies. And I think it's worth noting the ARCs are still, these are crashing onto the field, right?
Exactly.
Conjugated technologies, taking gene therapies and conjugating them to biologics. That's still only what, maybe 5 years old, John, 3 years old, really only in the last couple years has it been something bioanalytically that we're seeing, right? Sometimes we're kind of at the tail end of the drug or we pick up a drug development, right? We're not even looking at the R&D. That's why I love our little news and resources, because we're always seeing basic research, right? Oh,
Sure.
these are being done in bacteria.
Yeah.
That's
Yeah.
what we heard today. So I mean, this stuff is, I mean, we got 4 or 5 irons in the fire. Cell mediated gene therapies, your gene therapies that can be delivered as some sort of LNP or even just AAVs themselves, and then you've got this conjugation with the gene therapy. I mean, those are 3 huge buckets, John, that weren't even. But think about that. Bioanalytically, that wasn't even 5 years ago. The revenue streams for that were probably absent. Where
Oh,
today,
sure.
that makes up a
Yep.
chunk of the market, right? So that's how fast the market. That is a huge movement in the field. Like if I had, 5 years ago when we were talking, we wouldn't have been thinking about, oh, where's the market going? We probably would have said ADCs. Well, the ARC field is grabbing it, so that's
Mhm.
kind of staying true. But we knew that CAR Ts might be splashed onto the field. But let's face it, pre-COVID, the AAV field was there, but it was really for rarer, kind of hard to treat, and now it's mainstream, right? That is what COVID did with that one. It is popped off in a massive way. So that's my little diatribe for this one. But it's a fascinating study to think about just think about the percentage of what we call bookings or sales, even, John, 5 years ago versus today, and the
Yeah.
landscape has shifted dramatically
Oh,
into those three
yeah. I mean,
buckets.
as
Yeah,
we're increasingly dealing with, with gene therapy companies, and we go through an article like this, and it's like, well, I know who they are, because obviously we're we're familiar with them from work, and it isn't. I mean, because it's not just about the therapy, it's also, like you say, the effect of the therapy. So biomarkers become key. And so for me as a mass spec person, I wouldn't necessarily expect to have much involvement in this field, but we do, because there's there's biomarkers that if you're administering something, you have an effect, so you want to see how the biomarkers behave after administration, and is it doing what it's supposed to be doing. The other piece with the mass spec group is LNPs, that we've done a fair amount of work in that space too, where you can use the LNP to see where it's going and sort of test animals, etc., and to get an indication of what part of the body the LNP is going to as a surrogate for your therapy, etc. So, like I said, you think LCMS would have no involvement in this, but actually we do have quite a bit of involvement in this, and I think that then goes nicely into where else can we help in terms of given all the technologies that we have available, where can KCAS Bio help in the gene therapy space?
Yeah, no, you're that's a great segue there, John, to talk about how do we help gene therapies, and you touched on it. You went the mass spec is that's not new to us, John. We've been supporting LMPs for probably 10, 15, 20 years because again, LMPs go back to the 80s. This is not a new thing where people were making lipid nanoparticles to try and deliver gene therapies. That there's one of my mentors did it in Tennessee in like the 80s and 90s. Of course, it was a little different there. They were kind of just mixing it. They were taking advantage of the missile principles of lipids, right? That's how they formed them.
Sure.
But I digress, right? They just packaged it up and started delivering it into animals and things like gene guns. I don't know if you know what that is and stuff. So all these things to kind of deliver gene therapies. Well, you fast forward to today, you're right. We do a lot of mass spec work, and it's not just it's LNPs. We can do ASOs, right? You can do siRNAs.
Yep.
You can do all sorts of things on the mass spec, and then of course the real power of it. And then you mentioned the biomarkers. There's plenty of biomarker work there to kind of help in that space, but the protein mass spec has become massive, John, and that's really a big piece of it too, or the hybrid piece. So that's just when you think of gene therapies at KCAS Bio, the mind doesn't have to immediately start at PCRs. It's actually some of our strength, and I think we have a lot of strength in this space, is the mass spec space.
Sure. Yep.
But then if we pivot over to the next bucket, I'm going to save the molecular bucket for last because I think that's where people's minds go. It's actually, and I don't know the actual stats on what we support. That's an interesting thing. I don't wonder what exactly, what the breakdown is, but we know I can think of at least a half dozen LNP type clients we support, right, John? And then maybe even three to four of the ARC or conjugate, right? I can think of at least a couple there. In the cell-based gene therapy space, which is traditionally a flow method, right? So those are those CAR T cells, macrophage cells, whatever sort of chimeric cells they're engineering, NKs are splashing on the market, and we help with all those. I think we have maybe at least a half dozen active versions of that, and probably many more in our flow site out in Philly. So they got several there. I think we have the ELISpot can be used as well in that space, and I believe Leon might have a couple that they're supporting that way. So there's a high number of what we'll call cell-based gene therapies that we support. And then you get into the true, the AAV space where people start looking at the actual RT, the RNA itself, and that we have at least probably upwards of a dozen clients that we're looking at for biodistribution studies of let's call them viral or non-viral based vectors. So that's the round out, right, John? You got mass spec work, which is expensive for LNPs and any sort of silencing RNAs or maybe even the oligospace ASO, morpholinos, all of those, the biomarkers across all of our platforms, and then your cell-based gene therapies. That's going to be more flow and some ELISpot, and then your these viral, non-viral studies as well as traditionally a PCR-based assay, and we do that extensively. So, John, we've got a lot of irons in the field that we can support, a lot of tools in our tool chest for supporting gene therapies. And I'll kick it back over to you, but we definitely got a lot of, as Lawrence Goodwin likes to say, horsepower for supporting gene therapies.
I don't have a lot more to add. I just thought it was really important that this is something that if you have a good suite of bioanalytical technologies, there's a lot of support that you can provide for this field. And
I think to our audience, to our business development directors, to clients, to anybody who's listening, when we think of gene therapies or cell-based gene therapies, this doesn't mean it's just PCR and flow. But it's very important to understand that the mass spec and even we're doing hybridolizes. I should mention that. We're doing a ton of those. So that's a big part of And maybe John will take a minute. You were gonna, we hosted the AAPS Summer Scientific Forum, and we were hoping to maybe squeeze that in. Do we want to take a minute and I can share
Sure,
a little bit about that?
absolutely, yeah.
Because
Yeah. I mean, I
yeah,
think as regards this main topic, we're
yeah.
probably
So we're
close to
wrapping up the
done.
main
But
topic, and we're just going
yeah,
to take a few more minutes of time because we had a real exciting event. If those of you that don't know it, there's now 3 AAPS events, and that's the American Association of Pharmaceutical Scientists. It's probably the largest, right? We got RIB as another counterpart, but it's not as big. The workshops and regulatory issues of bioanalysis, but AAPS, they have their what they call the Pharm 360 meeting. It's their general meeting. It's their largest meeting. That's an October-based meeting. They have their NBC, which is the National Biotech Conference. So the larger AAPS has everything from CDMOs, right, GMP, to traditional GLP CROs such as us, so bioanalytical CROs, analytical CROs, pharmacists, students. It's massive, right? 5,
yep,
7,000 people.
yep,
The National Biotech one, the NBC, that's a little more concentrated in biotech, so it's much more bioanalytically focused, right? We like that one a lot. Tends to have more biologics, but it's still got a smattering of everything else. Gene therapy, the field evolves, and that one usually has a little over 1,500, a couple thousand attend in that one. Attendance is up in that one, and then so this was the second year of what they call the SSF, the Summer Scientific Forum. So there's 3 meetings. There's one in the fall, one in the spring, and this summer one. And this summer one has a bioanalytical track and an analytical track. And it used to be the Land O'Lakes, but they've now merged into AAPS. So it's the second year of this one. It's quaint. It's only about, this year it had about 150 people, but it was incredibly hard hitting, John.
mhm,
Like lots of good science to it, right? It was a lot of industry. It was very industry driven. So of the 150 people, probably 40% are vendors, right? And then
sure,
the
yeah,
other
yeah, yeah, yeah,
60% were large pharma. So it was it wasn't, I think I only ran into maybe 2 students there, which is a little bit unusual, lower number, although we love AAPS. I'm not against students, but it was this, because it's summertime, I think there are less students, who are traveling. So it was held in Kansas City, the second year of it. We hosted, when I say we hosted, we had a couple speakers, and I was a moderator. I was part of the planning committee. I've been working on this for months. And so Don Duffield talked about ADCs. We had plenty of clients that we invited that came in and talked about immunogenicity of ADCs. We had one talk about ARCs. And then Steve McCarthy of our Philly site came and talked about isoplexis and proteomics on that system. And he gave a really in-depth, man, that system's very powerful, John. We might have to have him on. That thing
sure,
does a lot of the clinical assessments to help with understanding, because this thing could do like 40 biomarkers on a single cell. Well, you generate all that data, you got to do something with it. So
wow,
there's some cool, exactly. He showed
yeah,
some great stuff. He actually ran out of time. I did a nice little panel. And then more importantly, John, we hosted, so we bussed everybody from the conference for dinner one night at our facility. You got to tour our facility. So about 65, 70 people took advantage of that opportunity. They came and took a tour of our 70,000 square foot facility, 4th largest in the company. And then we walked over to our K-State Olathe campus, which is about 0.3 miles away. Some took a bus, went over. It was a beautiful night. It was only about 75 degrees. We got lucky for July weather. And then we had a reception with barbecue dinner. And it was just a wonderful event. Lots of smiling, lots of talking. Sometimes you hold these events and sometimes they look like high school boys on one side, girls on the other, right? None of that. At one point it was magical. I know there's some photos on LinkedIn, but at one point I kind of, I'm kind of hosting, right, John? I'm running around trying to make sure everything's going well. And we lined up about 15 or 16 KCAS members volunteered to help give the tours, because we gave them in groups of 2,
sure, yep,
about 10 or 15 per group. So I want to thank that team, the executive team. We all showed up. It was great. But everybody got in, everybody got through the line, look up, and there's just a sea of people just talking and smiling, John.
good.
Didn't see
That's
anybody looking at their
fantastic,
watch,
yeah,
none of that. And then eventually we almost forgot we had to go back. That's how great it was. Nobody was coming up. Where are the buses? This and that. I had to make a little announcement, and people were generally, because everybody's coming from the city, right?
sure,
So we
yeah,
did something different. We took them out a little bit outside the city, and the campus there, the reception was on like a lake. So half the people were outside, half were inside. It was really great, John. So I mean, very proud of the organization, proud to have that many people come through our facility. But there was some science in there, wasn't there? There was a reason I was going.
Yes.
I kind of forgot where the science was, but it had some themes around some gene therapies were part of the talks, and just that's a wave. But I thought there was something I was going to talk about gene therapy wise, but I kind of forgot about it. But John, it was a wonderful event. I want to thank AAPS for allowing myself to participate in that. Little did I realize that when I signed up for stuff, John, I got to really look at the fine print. So I've got at least one more year as the chair of or co-chair of the critical reagents. So it's a 3-year tour there that I've been on 2 years. So I've got one more. And then there's a good chance I'll be heavily involved in next year's, because the people so I was a part of the bioanalytical committee, so the planning committee there.
Mm-hmm.
There, hats off. I cannot believe how much effort goes in. It's all volunteered. Every one of the conferences I mentioned, there's a chair, a vice chair. So there was 2 people there. And then we had the bioanalytical chair and vice chair, and the analytical chair and vice chair. And I was part of that bioanalytical committee. So next year I plan to maybe participate. We'll see how it goes. Maybe I move up a little bit, but it was, I was kind of cranky a little bit, John, right? The last few weeks I've been a little stressed, but just because it's taken on that extra kind of just capacity to help make sure this goes along smoothly. And it did, John. I painted a better picture. The weather was great. The food was great. Nobody, not one complaint really. And so it just went off without a hitch, which isn't always the case. But I want to thank my team and even AAPS for helping to make that work. We had a lot of planning. So I don't know if you got any questions for me, John, but it was a wonderful evening. The talks themselves were, again, I stayed solely on the bioanalytical track. Great stuff around ADCs and ARCs and the immunogenicity and PK. Boy, there's a lot of talk about how to do the PK for those types of drugs. Kind of the 1.5 method was talked a lot about, right? The power of hybrid. So I don't know, John. I know we're getting a little bit long here, but it was, you could hear it in my voice. It was such a great thing, and it was cathartic too. Boy, was I relieved after it was done, and but still, it was a very special event. At least I thought it was, personally and professionally. It was just wonderful.
Yeah, meantime, all I was trying to do is think straight because I was getting over COVID. So
yeah, that didn't help, John.
Definite contrast between the two.
It didn't help. I guess I stayed negative, but yes. I definitely caught, there was something going around. I actually stayed negative, knock on wood, but I definitely caught. I was a few days before the conference. I was feeling fine by the time the conference went around.
Well, actually, my wife has stayed negative all the way through this.
Isn't that fascinating?
Yeah, exactly. yeah. And I was rough.
Well, anyway, I mean, it wasn't just you, right? There was like 3 or 4 others who were positive, and then there were 3 or 4 like me that stayed negative. You just wonder what that all means. But
I don't know. Anyway, so I think that's it. I think we can wrap up now, and
yeah, John, maybe some parting stuff here. It's a little unusual that for those that don't, today's a Monday. That's why we're a little off. Usually we're leading into the weekends, but I don't know, John. I certainly, the summer is bombing by. I have not been cooking as much as I like, in part because I've been doing these volunteering for stuff. But I don't know if you want to share any weekend plans. Mine is all focused on dialing in my son going to college. I'll share first. We went and had his orientation at his fraternity here. He's a freshman going to Kansas University, and he rushed last year, so he's not living in a dorm. He's going right into a frat house. And so they had the parents over this past weekend, and we had a little barbecue. We met he doesn't have a cook, John. He's got a chef.
Oh, wow.
We got to see his fraternity house, which is more like a dormitory / like kind of looks like a Marriott courtyard, if you know what I mean. He's got an unbelievable house that he's going to move into. It's couldn't be prouder of him. And the house is unique in that it has no neighbors. There's a road on one side. He backs up to the football stadium in the backyard. You can see KU where this year they're redoing it, so that's a little bit of a bummer. They're going to have to play some games at Arrowhead and Sporting, but next year he'll back right up to the football stadium, which is that's a basketball school, but football is getting much more popular. And then there's a parking lot, like there's like a building, a faculty building, and or a university building in a parking lot. So he's got no neighbors, John. That's what I'm going at. I can't believe there's like 65, 70 kids living in this house. He's got a dorm mom who lives there the whole time. She's with them, and she's great. She's been there 11 years. The chef, he lives down the street, and then they got a senior member, a guy who makes sure everything's kind of working, and he lives right around the corner, so there's some safety there. But I mean, I don't know, John, I couldn't believe, I could see why he was so excited. They got a sports court out back. They got like a fire pit, an outdoor bar with a grill. I mean, it just looked like a Courtyard Marriott. I'm serious.
Sure.
It looked, going into his house this is not the Animal House sorority house type place, John. It was super clean, super, just and then it was all about academics. He is in a fraternity that had been known for having a good time, but over the last like 4 or 5 years, they've implemented some academic excellence. So there's still the thought of having some good time. Clearly, they look like they have fun, but now they're really focused on their GPA, and they've got a GPA of like, the house average is like a 3.4 or something.
Okay.
So he's expected to study, and I mean, it's a good mix of work hard, play hard, but you want a little crazy and a little wild. But most of it's there to learn. It's just a spattering, right, John? You don't want too much of those two things, crazy and wild. Exactly. But he's got the right mix. Sorry, that was a long thing, but again, I'm really a little bit emotional too, having the son leave the house here. I'm sure you've been through it. The wife is she's definitely, kind of bracing for it, and so am I. I'm going to miss him. But that's tying up my summer. And then I got to go to Lyon again, and we'll talk about that. I got to make my second trip to Lyon before this podcast.
See, we're through the other side, so we're happy to have the house to ourselves, etc. And we're firmly in reorganization mode. And my daughter's bedroom has now become like a office / relaxing room, and bookshelves and stuff have gone up, and we've just been doing a lot of that kind of thing. But actually, for me, as I mentioned before, I got COVID 2 weeks ago, and that's taken out a lot of time. But I'm sort of trying to get myself straight before I go to the UK in late August. So I'm spending a couple of weeks there. So actually planning that has been a ton of fun, and figuring out where it's all covering a lot of ground in terms of trying to see my sisters, both of whom live well apart from each other, and also doing some of the stuff I want to do. So that's really my focus at the moment.
Yeah, super exciting to be able to do all that, John. I'm sure you'll be seeing what's your favorite band there. Oh, you think I'd know?
I'm seeing the Chameleons.
Yeah, there it is.
I've seen them in a couple of weeks in Boston actually, and then I'm supposed to be they're playing a small music festival in Middleton, which is their hometown. And that happens to coincide with when I'm in the country, so I'll go and join them for that, because it seems rude not to.
Yeah. Your summer's a lot more adventurous than mine, and it's a small sacrifice here to kind of stay. I got to go buy a whole bunch of dorm stuff for him, or
sure.
Yep. But yeah, no, that John, that sounds a lot more enjoyable and adventurous than our prepping for. But again, small sacrifices, and then he's going to be out of the house, John. We haven't even thought about that. That my wife's not quite ready for that discussion, but it is something I'll start thinking about. But that's awesome, John.
Yeah, have to.
I'm glad you're feeling better. I'm excited you'll be able to go visit and see your family and
sure.
all that.
Yep. Okay.
Well, I gotta say, I haven't been making any pizza. My son's still working at the pizzeria, so no pizza in my world. But I can't go without saying it. And I actually, when he leaves on the 23rd of August, and so don't be surprised that in somewhere in there, my uni pizza oven. I've been thinking about that. I can't wait to start making pizzas again. But right now, he brings home a pizza like 3 or 4 days a week, John. I don't need to be making pizza.
Yeah, absolutely not.
Yeah. Anyhow, I can't go a podcast without not mentioning pizza. Yeah. Lately, I've been making some great marinara sauce. That's been my summer.
I haven't been cooking at all, but I'll pick that up sometime.
Yeah, no, like you, the last few weeks have been a blur, right? But I had been doing some marinaras because of the tomato season and all that. But John, we can wrap it up. We'll say goodbye to everybody. John, thanks for joining us today.